Hello everyone, in this video I'm going to complete a lesson on the trunk or the torso. So far we have looked at the muscles of the torso, we have looked at the stabilizer muscles, we've looked at some abdominal muscles, and we've also looked at the intercostal muscles, so the ones that move your rib cage and help you to breathe. Then we moved into talking about the bones, pretty much looking at the axial skeleton from vertebra C1 down. Of course, the torso muscles that we've discussed originate and insert on different parts of the axial skeleton within the trunk. I would like to continue talking about the spine, so the bones of the trunk, and a little bit more about how they join together. And then we're going to zoom in and look inside of those bony structures. And we're going to then focus on the nervous tissue within the trunk as well. So where we left off talking, I was discussing just the different ways that vertebrae articulate or form joints. And so there are a couple different ways. First of all, we have these articular processes. Okay, so here we can see the superior articular process of an inferior vertebra. Up here, we can see the inferior articular process of the superior vertebra, and we can see that they join together in this way. Of course, there are pairs in them, so this is happening on both sides. To look from the posterior side, this is like coming out of the screen at you. This is the spinous process, which we can see right here. Okay, and of course we can see the articular processes, and they are covered with these discs of cartilage. Okay, so that is one type of cartilage. We're going to learn a lot more about that when we talk about bone tissue. But this pretty much protects the bone here so that there isn't a lot of rubbing and damage on the bone itself. The movement facilitated by this shape of joint, this type of joint, is pretty much, the articular processes can kind of slide along one another. Here we can see extension of the spine, so kind of standing up straight, and here we can see sliding in the other direction, so flexion of the spine, or like, doing crunches, bending over. And so a lot of that movement is facilitated by just the shape of these articular processes. What we can also see is that these intervertebral discs are really important in this spine movement as well. We can see that for the most part, these are cushions, but they do kind of flex a little bit and definitely hold the bodies of the vertebrae apart. Okay. I want to talk a little bit more about these intervertebral discs. They aren't made of the same tissue as this. This is hyaline cartilage. The intervertebral discs are made of a structure that is much more rigid. It is, strong against damage, and that is due to the additional fibers within it. Okay, these intervertebral discs have an outer ring of fibrocartilage, so super, super strong. And inside there is a fluid, so that's what this is here, that is called the nucleus pulposus. So a little bit more about cartilage. There are 3 different types of cartilage, all of which are considered to be connective tissue, which, as you know, that means that there is mostly non-living structures, so mostly protein, mostly fluid, as opposed to cells themselves. The different types of cartilage are classified according to their structure, of course. Hyaline is by far the most common throughout the body. Elastic is a little bit stretchier, so you can think of like your ears, right, that you can pierce and see and everything. That is made of elastic cartilage. And fibrocartilage, what makes it different is that it has a ton more fibers. Okay, so what we're seeing over here on the right is some histology of fibrocartilage. We can see that most of this pink here is actually a rigid matrix of collagen, a couple other types of proteins, and that pretty much traps water molecules and gives cartilage this kind of like squishy texture. Okay. In fibrocartilage in particular, there's even more collagen than there is in the other 2 types of cartilage. And what we can also see is that there are these structures. And what this actually is a little hole right, called a lacuna or a lacunae for plural. And this is essentially a hole left during the process of building up cartilage in the first place, and filling that hole are the cells. Okay, so here we can see site, that means cell, and chondro means cartilage. And so chondrocytes are just the cells that maintain the cartilage. They receive signals, they might try to repair the cartilage if there's damage. And so they just kind of live in these little holes. And so we can see that most of this tissue is made up of collagen. So all of this would be collagen in here. And interspersed, there are these little holes with the chondrocytes inside. Now, the thing about cartilage is that it has a very poor blood supply. So unlike most connective tissues, which are very vascular, and in fact, blood is a connective tissue itself, fibrocartilage and the other two types of cartilage don't receive their own blood supply. So blood gets really close, but never actually perfuses this particular tissue. And so that has some implications. Any damage to the fibrocartilage, so these intervertebral discs, is going to take a really long time to heal because you need blood, you need resources to go to this tissue to actually rebuild the damage. Okay. Hold that in mind. We'll come back to that here in a moment. Something else that I want to discuss now that we're looking at the spine is that in between these two articulated vertebrae, there are holes. Okay, we know that there is an intervertebral canal that is pretty much a canal formed by multiple vertebral foramina of all of these different vertebrae. And so that extends vertically, but then kind of coming out to the sides, there are these lateral holes that are only formed when the vertebrae are articulated together. So only when it's a spine, not an individual vertebra. And these holes are called intervertebral foramina. And this is where something called a spinal nerve is going to exit. Okay, so what we can see here is the spinal cord, which is within this canal. And in order to actually deliver a message from the brain, in order to send sensory information up to the brain, these spinal nerves must exit the spinal cord and deliver information or either to or from the body. Okay. So these spinal nerves are named in largely the same way that the vertebrae are named, and their name is derived from an adjacent vertebrae. Okay, so we're going to start with the cervical vertebrae. There are, as we know, 7 cervical vertebrae, C1 through C7. Here we can see kind of a cross section through those vertebrae. This back here is the spinous process. Here is the body. And so again, this is a section through. This is the spinal cord. And so what you can see are these nerves that actually exit from the spinal cord out to your peripheral tissues. And so we name spinal nerves in the cervical spine, C1 through C7, based on whatever vertebra is underneath the spinal nerve. So this first spinal nerve that exits up here, that is over top of it, is superior to vertebra C1. And so we call it nerve C1. Okay. This one here is over vertebra C2, so we would call it C2. C3 is over C4, and so on and so forth, all the way down to C7. Now, it gets a little tricky after this. There are, in fact, 8 cervical spinal nerves. So even though we only go to C7 in vertebrae, we go all the way to C8 in nerves. And so C8 exits inferior to vertebra C7, vertebral prominence, and from there down, the nerve is named for whatever vertebra is on top of it. Okay, so here we see nerve T1 is just inferior to vertebra T1. Okay, just for example here, this is vertebra T8. Nerve T8 would exit underneath. Okay, here All right, this would be nerve T12. This would be nerve L1. Okay, so it is fair game for an exam for me to ask you what spinal nerve would exit superior or inferior to, vertebra L4, and the superior one is L3, the inferior one is L4. Okay, so definitely take some time to practice that. For the cervical, nerves come out on top. Everywhere else, nerves come out on bottom. Okay. And so now we have a pretty good understanding of the bones, the muscles, and the nerves in the region of the spine, so the dorsal side of the trunk or the torso. And so I want to talk for just a moment about some damage that can affect these structures, right? And that's because they're all so very close together. Okay, so there are a couple different types of conditions that would lead to back pain. Right, so lots of different things. The two that I want to bring up are, first of all, degenerative disc disease, and essentially, a healthy fibrocartilage disc is nice and thick. It's pretty tall, really strong. But there is a condition called degenerative disc disease where the fibrocartilage itself starts to break down. So instead of being this thick like a healthy disc, now the discs are much narrower. And so that does a couple things. First of all, it doesn't hold the bodies apart as much as it should. And so if this individual flexes forward, so bends forward to tie their shoe, now this body might rub up against this body, and that can lead to bone spurs, okay, and other painful consequences. Okay. Also, because it isn't holding these bodies together very well, or not holding these bodies of the vertebrae apart the way it should when it is thicker, essentially those intervertebral foramina are going to get much smaller. And so the spinal nerves that we just talked about that have to exit through those holes all of a sudden can become pinched, they can become squished. And so that affects the ability to send messages from the brain via the spinal cord. And also it can be a very painful situation. Okay. Similarly, the intervertebral discs, when they are under a lot of wear and tear, they have the ability to become herniated. So normally the nucleus pulposus, the liquid, is held within the intervertebral disc, and this gives it part of that sponginess. On the outside is fibrocartilage, which is really rigid, but if you are, in a car accident or other type of accident situation, or, a lot of intense wear and tear on your spine, like, playing football or something, or if, you're obese or otherwise have, kind of weaker fibrocartilage discs, that nucleus pulposus can actually kind of squeeze out to the side here. And so we can see in this image that the spinal cord and the spinal nerves are so very close to the fibrocartilage discs that once again, they can be pinched, right? This can, lead to pain, tingling down, in this case, the front of the leg. That's where these spinal nerves will be going. And, maybe even the inability to control those particular structures. Okay, so both of these can lead to nerve damage as a result of something going on with these intervertebral discs. I also want to point out that over the course of a day, and for all of us, these discs get narrower and narrower. They kind of get squished with the weight of your body and the pressure due to gravity. And so if you measure your height, at the end of the day, it's actually like a half an inch shorter than if you measure at the beginning of the day. And so, you can start to lead to these issues like lots of soreness and everything as well in that case. Okay, For both of these conditions, of course, there are a variety of treatments, but I bring this up here because your stabilizer muscles, your multifidus muscles, your erector spinae muscles actually have the ability to, essentially pull this spinous process down, which essentially pulls the body up. And so what your stabilizer muscles can therefore do is take a little bit of the pressure off of these poor intervertebral discs and therefore not kind of squeeze them out to the side like this, or not have the bodies rubbing together. Okay? So having these stabilizer muscles be really strong, right, and ultimately activated all the time, this actually can reduce the pain, reduce the discomfort of conditions like degenerative disc disease and herniated discs. Okay, so these stabilizer muscles are really important in alleviating some of those symptoms. A lot of times, as you can see here, back pain is associated with weaker stabilizer muscles that aren't taking the pressure off of these discs. And so, of course, then you ask, like, how do you strengthen these things, right? And so more or less, you don't go to the gym and lift weights and everything in order to strengthen these muscles. That's just not how these are built up. Instead, any kind of like balancing exercise or core exercises are inherently building up these muscles. So anything where you're like standing on one foot, or like with this Swiss exercise ball here, you have to stabilize your core as you are balancing. And so like every tiny little movement as you are balancing on the ball is actually recruiting these multifidus muscles, right, automatically to remain balanced on the ball. And so if you could, sit on one of these at your desk instead of a normal desk chair, if you can, do other kinds of balance exercises and core exercises, you are strengthening those muscles. And so if you do happen to develop some kind of condition like either of these or other kinds of back pain, you already have the muscles engaged. And so hopefully the consequences won't be as severe. So a little bit of a tangent, but of course, very relevant to all of our lives, especially if you were going into PT and OT. These types of exercises are definitely helpful for patients with diseases like this. Okay. So back to these spinal nerves. Yes, they can be damaged with these disc disorders, but what do they do normally? Okay. In fact, we have 31 spinal nerves, right? If you count that up, we have 24 vertebrae, right? 25 spinal nerves that are named according to a vertebra that is adjacent. And then the last 6 are exiting via the sacrum and the coccyx. Okay, so ultimately, these 31 spinal nerves, what is their purpose? They innervate specified regions of the body. And so what does that mean? Innervating is both sensing, right? Like what is going on in my skin, what is going on in my muscles, as well as supplying motor control. And when I say motor control, I mean action potential down a neuron, releasing acetylcholine onto the motor end plate, and therefore the muscle is triggered to contract, right? So your brain decides, I want to move my arm, and that message is sent down the spinal cord and then ultimately out the spinal nerve in that particular region. We have a really good understanding of what spinal nerves are responsible for innervating different parts of our body. In fact, we can map out what are called dermatomes, right? So derma skin, and tome, like anatomy, is essentially like named after the process of like cutting up structures to learn what's inside. So these are skin cuts, right? And so each one of these codes here, right? This is spinal nerve C4. This is spinal nerve T1. And so when those spinal nerves exit out the intervertebral foramen, they are going to go to some nearby place, or, even sometimes like down front of the leg or something. And that spinal nerve is going to innervate, right, this particular strip of the skin and the muscle, beneath that. Okay. So the skin, the fascia, etc. And so the reason why this is so important, right, these dermatomes, these mapping of all of the spinal nerves and what they control, is that we can actually see or predict what spinal nerves are damaged, what spinal nerves are infected, for example, by looking at what different dermatomes are experiencing. So for example, if you contract a shingles virus, okay, that usually presents as like these patches in stripes in your abdomen. I've also seen it like down the arm here. And so these patches, and essentially we can isolate where exactly is the location of this infection, because we know what dermatome is infected. So here, the spinal nerve T10 is infected if there are patches down here. Okay. Also, if there is nerve damage, and suddenly you have a tingling sensation along this stripe of skin, or you can't feel it or whatever, that automatically tells us like this is the spinal nerve that is affected. Okay. We're going to look at the spinal cord here soon, but I want to point out that spinal nerves split or bifurcate really soon after they exit the spine. Okay, and so this here is the spinal cord, so within that vertebral canal. Ultimately, the spinal nerve is what finally exits from the spine. As we can see in this image, the spinal nerve splits into two other branches. Okay, and so we can see here that this branch, right, so a bunch of neurons, is going to innervate whatever is in the back, right? So all of those stabilizer muscles on your back are innervated by this particular root, as opposed to this branch, right? These neurons travel around, right? They go around to the front of the body to innervate the tissue on the front. And so these two branches, we of course have names for them. The dorsal ramus, of course, is going to go to the dorsal side, right? All of those stabilizer muscles on your back. And the ventral ramus is going to go to the belly, to the ventral side. Okay. And I'll come back to that in just a moment. There are also other branches here, and I'll show them to you on this side. These branches are containing neurons that are part of your autonomic nervous system. So essentially, these particular neurons are going to release neurotransmitters onto the surface of your heart or your lungs or your stomach. Okay, so these are all more like automatic functions, as opposed to consciously thinking, I want to move my arm, and your arm moves. Okay, so these branches here are essentially communicating with your visceral organs, heart, liver, intestines, whatever, via this trunk right here. Okay, so this, and actually this over here is actually part of your sympathetic nervous system, in fact, so it kind of ramps up your body to run away from a bear or some other fight or flight response. And so these branches that relay information to the sympathetic chain of ganglia, which of course is parallel to the spinal cord, but outside the spine, they're called communicating ramus, or with an I instead is plural. Okay. Okay, so once again, the spinal nerves divide into rami, or singular ramus, as we can see at the top. Visceral commands of the sympathetic nervous system travel via the communicating rami to the sympathetic chain of ganglia. Okay. Voluntary information and conscious sensory information is going to be relayed to the back via the dorsal ramus and to the front via the ventral ramus. And so I said I wanted to come back to this ventral ramus right here. And so the ventral rami, multiple ramuses, are going to be really important to us. Some of them, or rather depending on where they are, depending on what nerve they're coming from, they are going to get special names. Okay. First of all, in the thoracic region, right? So those 12 vertebrae all articulating with two ribs, those spinal nerves, ventral rami, are going to travel inferior to the rib itself. So that little groove that we looked at in the lab, that is where the nerves are, along with the blood vessels. And what nerves those specifically are the ventral rami of the spinal nerves in the thoracic region, so T1 to T12, and we call them the costal, another word, rib nerves. Okay. So in the lab, we actually have a model that shows this pretty well. And so I'm going to bring back this model a few more times throughout this talk. Again, here's the spinal cord. We can see these are the transverse processes. This is the spinous process. So we're looking at this model from the back. We'll talk more about these in just a moment, but this here is the spinal nerve, and it is going to branch pretty much immediately. Back here, this is the dorsal ramus, right? We can see that it would be going to muscles that would be back here. So all those stabilizer muscles. Okay. The other branch is pretty much going out, traveling along a rib, right? So this here would be a rib. These are the ventral rami. And we can also see other branches, right? So this one and this one is going to that sympathetic trunk here. These are the communicating rami. Okay. So again, we're going to talk a little bit more about what all of this stuff is here in just a moment, but I want to point out the ones going to the back are dorsal, ventral, and communicating. And this thing that's parallel here, this is the sympathetic chain of ganglia. Okay. This is an image that is kind of mapping this out a little bit more. Sorry, my bird is excited here. This is the spinal nerve. It splits to dorsal and ventral rami. And here are the two communicating rami as well. Okay. As I said, the ventral rami of the thoracic spinal nerves are ultimately going to be called the costal nerve. So you can see each one of these is going to be just inferior to one of the ribs. In the rest of the spinal cord, or for the rest of the spinal cord, the ventral rami are going to form something that's called a plexus. And a plexus is this complex interwoven network of nerves. So essentially a ventral ramus from this spinal nerve, a ventral ramus from this spinal nerve, they're going to come together and then they'll split apart, right? And they'll come together and they'll split apart. Essentially it forms this rich network of nerves. And so what this does, it's kind of a fail-safe, right? Even if you damage, this right here, you still have the information getting out, in this case, to your arm via nerves in the rest of this network. Okay, we can name the four main plexuses according to where they are in the body. Remember, I told you that those regional terms would be important, and really they just keep coming back. In the cervical region, go figure, we have the cervical plexus. In the brachial region, we have, or rather, the brachial plexus sends information to the brachial region. And remember that that is the upper arm. Okay. The lumbar plexus is literally in the lumbar region. And the sacral plexus is here. And it is made up of nerves that are exiting from the sacrum. Okay. In the next unit of this class, we're going to talk about individual nerves that arise from each of these plexuses, but for now, just know where they are and what they're made of. Okay. So at this point, we've talked a lot about the different spinal nerves, right? We've talked about how to name them. In this diagram of the nervous system, the spinal nerves are here in yellow. We can see, for the most part, the ventral rami are going out to the rest of the body. Again, in the next section of the class, we're going to name like this nerve, and we're going to name this nerve, and like all these other ones, okay? Before we get there, right, let's kind of zoom out a little bit and talk about the flow of information along these spinal nerves and ultimately to something that's called the central nervous system. Okay, so general flow of information. In the beginning, right, maybe you prick your finger, okay? That information is going to be sensed on receptors at the periphery of your body. Okay, so in this particular dermatome, whatever. So you prick your finger. We know that that is going to stimulate receptors on a sensory neuron. You get depolarization in the dendrites. If the stimulus is strong enough, threshold is achieved, and therefore an action potential is going to be generated and fly down a neuron within one of these nerves. Okay, so it's going to go in that direction. Okay, again, these are sensory receptors. Different receptors detect different types of information. Okay, so this is all information that is being detected in the peripheral nervous system because it is all in the periphery of the body. Okay, not only is information about the skin and the muscles being detected, but you also take in information from your viscera, your heart, your lungs, your liver, as well as special sensory receptors. So your sight, hearing, taste, all of those things. So it largely works in the same way. We are going to come back to this at the end of the term, and we'll talk about this in A&P2. For now, let's just focus on these somatic sensory receptors from the periphery of your body. Okay, so once the action potential has been generated, it travels along these spinal nerves and finally should enter into the central nervous system. The central nervous system includes the spinal cord here, right, as well as the brain. Okay. In the entire central nervous system, brain and spinal cord, there is this integration of information. Okay, so information about pricking your finger or getting a splinter or whatever is going to be sent to the spinal cord. If you need to immediately pull your hand away from, maybe something that's hot or, otherwise, oh my gosh, like really need to get your hand out of danger, your spinal cord can actually make that decision. It integrates information like, when I experience this level of heat, chances are my hand is on the stove and therefore I need to get my hand out of there. Okay. Also, information is sent up to the brain. Okay. And the brain can actually do a lot more processing of this information, right? So like under these conditions, like I remember this one time this happened when I felt this way, and therefore this is how I should respond, right? So your brain is going to integrate your memories and all sorts of different sensory information from throughout your body. It's going to make a decision, and it's going to send that command, that motor command, back down the spinal cord. Okay, and out to the peripheral nervous system. Okay, so for us, for now, right, this information, right, maybe given this stimulus, maybe you want to tell, right, maybe your biceps brachii to contract and therefore remove your hand from whatever is damaging it. That information would then exit out from the appropriate region of the spinal cord. Action potentials traveling down these spinal nerves, right? And finally, commanding, right? Telling the muscle, contract, remove your hand from the danger. Okay, so this motor information, right, is also sent down the spinal cord and is going to exit out the spinal cord via those spinal nerves to go to its target tissue over here. Okay. As I said, there's also autonomic nervous system information that travels along the spinal cord as well. This is an A&P2 concept, but of course we've seen a couple of the structures that are involved in sending these messages. That is the communicating rami and the sympathetic trunk of ganglia. Okay, so Neurons themselves can be classified according to where they're taking information, right? Like what information they're actually carrying. When your skin detects, whatever temperature, that information is carried along a sensory neuron. All right, notice that we have about 10 million of these sensory neurons in our body. So we are constantly assessing what is going on in our skin, in our viscera, in our muscles, in all of these different places. Okay, so sensory neurons carry information to the central nervous system. Okay. Another way of looking at this, here we can see this guy stepped on a bottle. That really hurts, so nociception. And so the action potential travels up this sensory neuron and finally into the spinal cord. Now, in the central nervous system, there are other neurons that have essentially been wired in such a way that they remember what happened before. They know that this particular stimulus needs to have this particular response. And so there's a lot of communication via neurons back and forth in your brain and even in your spinal cord itself about the incoming sensory information. And so all of those neurons that are talking back and forth, trying to come up with some kind of decision about what to do, they are called interneurons. And note that we have about 20 billion of those in our body. So we really think that it's important to integrate information as much as possible. Okay. Finally, motor neurons. In comparison, hardly any, so 500,000 of them in the body. These are going out, or rather taking information out of the spinal cord, right, out of the brain, and to the rest of the body. So here we have all these little neuromuscular junctions, right, telling our muscles what exactly to do. Okay, so there are only about 500,000 of these, meaning that we value thinking about our decisions much more than actually executing those decisions. Okay, right. As a human, it's very important to remember what's happened before, to look for patterns, to really assess the situation before you just randomly kick out your leg and end up putting yourself in even more danger. Okay, so sensory neurons carry sensory information to the brain. Motor neurons carry motor commands away from the central nervous system, and interneurons are talking back and forth within the central nervous system itself. Okay, so those are the three functional classes of neurons. Neurons can also be classified according to their structure. Right, of course, as you'd imagine. Unipolar neurons, these are generally those sensory neurons, and that's for the most part what I've been drawing on the board. Dendrites, a single axon, and maybe lots of synaptic terminals. What makes this a unipolar neuron is the fact that there is one single branch off of the cell body. Okay. So, right, cell body is somewhere along the axon. Bipolar neurons, right, bi2, have two branches off of the cell body. One branch is the dendrite, one branch is the axon. For the most part, these are the rarest type of neuron that we have in the body, and that is because they are only really present within your special senses. So we'll look at them a lot more at the end of the term. Finally, multipolar neurons have many different branches off of the cell body. So here's the cell body, and we can see dendrite, 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 dendrite. And of course, one single axon relaying information all the way down to the final destinations. These are the most integrative neurons that we have, right? So maybe over here we have sensory neurons telling us about the temperature. Maybe over here, sensory neurons telling us about what we see. Maybe over here, sensory neurons telling us, right, a memory of something, whatever. All of these neurons are either depolarizing or hyperpolarizing this neuron. And so in this way, the multipolar neuron is integrating information from this neuron and this neuron and this neuron, or even like from other parts of the brain, before it either generates an action potential all the way down, or it does not. Okay, so this is very like higher order integration, and therefore multipolar neurons are found in the central nervous system. So those interneurons, okay, taking in lots of different information before finally like making a decision here, and motor neurons. Okay, so maybe, you know, multiple different parts of, you know, the body, the brain, can decide that, hey, I should contract this muscle, I should move my arm in this way. And so any of those places can trigger an action potential and therefore command a muscle to start contracting. And so motor neurons, yes, there are fewer of them, but they are receiving information from a ton of different places and telling the muscle to contract or not. Okay, again, these are the three structural classes, and the structure is determined by how many branches there are off of the cell body. Okay. So what we are about to see is how information, sensory information and motor information, goes to and from the spinal cord. What we're going to see is that sensory information enters the spinal cord in one way, right, from one area. It's going to communicate with interneurons, okay, and some of these interneurons are going to go up here and deliver information to the brain. Others in reflexes, which we'll talk about more next week, are going to immediately synapse with motor neurons. Okay, so what these little balls here are dendrites. These are axon terminals. And so this is a synapse and this is a synapse. And so the interneuron sends a message to the motor neuron, which exits a different way and carries information out to the rest of the body. Okay, so that is where we're going here. Okay, also on this model here, right, sensory information comes in from the back. Okay, motor information goes out from the front. Okay, so let's add some names. Okay, this is the spinal cord. Okay, this entire thing here is a transverse section through the spinal cord. We already know the spinal nerve. Okay, so this is what would be exiting from the spine and then immediately or soon bifurcating into the dorsal and ventral ramus. Okay, note that spinal nerves carry both sensory and motor information. Right? So they're innervating, they are both stimulating a motor response as well as sensing information that is coming in to that particular area of the body. Okay, the spinal nerve splits, bifurcates into what are called roots, right? So we have a dorsal root. This is going around to the back, right? So your spinous process, the little bumps you can feel through the back of your skin would be back here. And over here, this would be towards your belly, towards the front of your body. Okay, so this blue neuron here is representing a sensory neuron. Okay, remember that sensory neurons are unipolar. So this here is the neuron cell body. And it's just kind of like hanging off of the axon. All of the neuron cell bodies of the sensory neurons are grouped together, kind of protected within the spine. Okay, and essentially like along the nice smooth nerve, there is kind of this like bulbous structure right here. Okay, this is a group of neuron cell bodies, okay, called a ganglion. In general, a ganglion is a group of neuron cell bodies. And here, this happens to be on a root, so one of the two branches of the spinal nerve, which is called the dorsal root. Okay, so this little bulbous thing right here is the dorsal root ganglion, a group of sensory neuron cell bodies that is really close to the spinal cord. Okay, the sensory information is therefore delivered into the spinal cord via the dorsal root. Okay, and here we can see it's going to synapse with an interneuron. Okay, another name for sensory information or sensory neuron is afferent. Right? I know this word is normally pronounced afferent, but I want to emphasize afferent, which is different than efferent down here. Okay, so a little ridiculous, but really important. These are opposite terms. Afferent goes to an organ. So in this case, the sensory neurons are carrying information to the central nervous system, as opposed to motor neurons, which are going to carry information away from the central nervous system. So E for exit, this information exits. It's this red neuron here. The neuron is exiting, or the information along it is exiting out the spinal cord and going away from the central nervous system. Therefore, it is efferent, also called motor. I'll come back to that in just a moment. That is important. Note that motor fibers, or motor neurons, efferent neurons, are going to exit out the spinal cord on the opposite side from the sensory neurons. Okay, these are going to exit and be bundled together to form the ventral root, so the belly side root. Okay, ultimately, the dorsal root and the ventral root join together to form the spinal nerve. And as we know, the spinal nerve will soon split to the dorsal ramus and the ventral ramus. Okay, both dorsal and ventral rami contain both sensory and motor information. The only structures that only carry one type of information, sensory or motor, are the dorsal and ventral roots. Okay. A nice way to remember which one is sensory, which one is motor, is with Dave. Okay. The dorsal root contains afferent information. All right, dorsal root, afferent. The ventral root carries efferent information. Okay, so knowing afferent and efferent will definitely help you to, use this little device. Also, afferent and efferent are not going away. Those are just generic terms meaning to something and from something. They will come back. The next structures that I want to point out in the spinal cord are white matter and gray matter. Okay, note that all of the synapses here are located within the gray matter, and all of, something else, which we'll talk about in a moment, is white matter. Okay. So in order to talk about gray matter and white matter, I need to talk a little bit more about nervous tissue in general. Okay. Remember that neurons are the functional units of the nervous system. That is, they are carrying messages, carrying information to different parts of the nervous system in the body. Okay, so those are the functional units, but they don't act alone. In fact, nervous tissue contains many different cell types that ultimately are there to support the function of those neurons. Okay, in general, these kind of helper cells are called neuroglia, or just plain old glia or glial cells. We, in fact, already know some of them. Okay, the Schwann cells are in the peripheral nervous system. Again, I'm very sorry, my bird is all worked up over something, and I have no idea what. In the peripheral nervous system, myelin sheaths are composed of Schwann cells, right? The Schwann cells are not, being depolarized. They are not, sending action potentials, none of that stuff that the neurons do, but they really do help the neurons to accomplish those goals. Okay, so remember here, this is a Schwann cell. Okay, there are two different types of myelinating cells. The Schwann cells, of course, are in the peripheral nervous system. So if we were talking about a neuron within a spinal nerve, we would be talking about a neuron myelinated with Schwann cells. On the other hand, in the central nervous system, in the spinal cord and in the brain, myelin sheaths are made up of cells called oligodendrocytes. A nice big word to impress your friends with. These look a little bit different than individual Schwann cells like these, in that oligodendrocytes have one big old cell body, and then they have multiple branches that can myelinate multiple axons at the same time. And so this makes sense because the CNS is just chock full of neurons, right? So billions of neurons in our central nervous system. And so one cell can myelinate multiple of those axons that are right next to each other. Okay, so again, these two cell types are a type of neuroglia. Okay. Let's kind of zoom into what is happening in the spinal cord. Okay, so this here is the spinal cord. This is the front, that is the back, so ventral and dorsal. And so we are going to zoom into this little part right here. All right, note that there's a little hole called the central canal. That's this right here. And that kind of like butterfly shape that we saw before is this here. That's the gray matter, and it's surrounded by white matter. Okay, so we are looking at the gray matter here and the white matter. So what I'm getting at here is that oligodendrocytes, these are myelinating cells. And so neurons, axons in particular, axons within the central nervous system are myelinated. Okay, so they are covered by these oligodendrocytes. Oligodendrocytes appear white because they are filled with lipids, right? And that's a great insulating material. And so when you put, hundreds, thousands, millions of these things together, all covered by this white myelin sheath, the tissue itself looks white to the naked eye. And so areas of the central nervous system with lots of myelinated axons, we call that white matter. Okay. And so in the spinal cord, the white matter is actually containing these myelinated axons, and therefore this is the region of the spinal cord that contains the structures carrying information to and from the brain. Okay, so what we can see down here is a section through the brain. Here we can see a section through the spinal cord, and so the dorsal roots, the ventral roots, okay? And in the white matter surrounding the gray matter butterfly in here, these are actually what are called tracts. So like bundles of myelinated axons carrying information all the way up to the brain. Brain integrates this information with tons of interneurons. And finally, motor decision is relayed back down and can exit to the part of the body where it is relevant. Okay, so white matter is where information is going up and down. Gray matter, on the other hand, that is where all the synapses are taking place. So it's not white because these parts of the cells, here we can see the cell body of this multipolar neuron, the myelin sheath stops so that synapses can take place. And so without so much myelin, this type of tissue is called the gray matter. And honest to goodness, to the naked eye, you can see the difference between white matter with all the myelin and gray matter without all of the myelin. Another type of glial cell in the central nervous system is called an astrocyte. Astro for star, cyte for cell. So these cells look roughly like little stars. So here, these cells look roughly like stars. Astrocytes have many different functions. First of all, they help to maintain the blood-brain barrier. So in this image here, again, a section through a spinal cord, here is a capillary, so that's a blood vessel. Usually capillaries are just, made up by these really thin layers of cells. Think simple squamous epithelial tissue. And the reason for that is that, we need a lot of different things to transfer over that skinny membrane really quickly. Now, of course, the central nervous system, we need to protect and be really selective about what is allowed into the nervous tissue and what isn't. And so there's an extra layer of protection. And here we can see astrocytes have these little extensions here that fuse together and are therefore adding this extra layer of protection. Okay, here in this image, this is the membrane of a capillary, right? So here are all sorts of different cells, and astrocytes, right, form this blood-brain barrier. Okay. Astrocytes also provide structural support. So here, again, the astrocyte has these little extensions that are securing an axon and just giving it a little bit of extra strength in addition to its cytoskeleton within. Astrocytes assure that the extracellular space, right, which usually has, right, lots of sodium and a little bit of potassium, right, and lots of other things, astrocytes make sure that potassium levels, for example, don't get too high. Remember, if potassium gets too high, that depolarizes the cell and makes these neurons fire a lot more easily and therefore a lot more. And so that's just one example. Astrocytes make sure that if potassium gets too high, it takes it into the cell. Similarly for nutrients and for different gases. Okay, so it just makes kind of an ideal environment for the function of the neurons themselves. We have talked about how acetylcholinesterase is an enzyme in the synapses that helps to break down and ultimately recycle acetylcholine. There are enzymes for all sorts of different neurotransmitters that do that, and astrocytes are an important part of that. So astrocytes can synthesize these enzymes and also can be helpful in reabsorbing some, just to make sure that at the synapse there's not stimulus with leftover neurotransmitters from a previous message. And finally, nervous system damage is extremely slow to repair. However, it's not impossible under the right conditions. And one of the ways that the nervous system can repair itself is with astrocytes. So astrocytes can help to form scar tissue after injury to the nervous system. It can kind of support these cells as they are regrowing and therefore regaining their functionality. All right, so astrocytes pretty much do it all. Microglia. Here we are focusing on this particular part. Okay, this is part of the central canal, which is right here. Or oops, sorry, not with the microglia. Pardon me. Before we get into the central canal, let's look at the microglia. All right, microglia are essentially the little Roombas of the central nervous system. Of course, with the blood-brain barrier intact, we don't get a lot of perfusion of immune system cells into the central nervous system, or at least that's been the general understanding for quite some time. Instead, relatives of our immune system cells migrate into the central nervous system very early on in our fetal development, and they are going to live, for the rest of their lives, rest of your life, in the central nervous system. And so these little guys here are the cells I'm talking about. Again, little Roomba cells, and it even looks like they have little brooms. And so anything that doesn't look quite right, anything that does belong there, right? So bacteria, viruses, whatever, any kind of waste products, right, are endocytosed via these microglia and therefore eliminated from the extracellular environment. Now, ependymal cells, the final type of glial cell that I would talk about in the central nervous system, these ependymal cells are lining what is called the central canal. Okay. The central canal is filled with cerebrospinal fluid, so kind of like a filtrate of the blood, helps to get all the good stuff to our cells and wash away the waste products just from, daily life, just like the blood would do in the rest of our body. And so cerebrospinal fluid needs to be kind of washed, you know, cycle through, washed away. And so it is circulated not with a pump like the heart, but instead with these little ciliated cells called ependymal cells. Okay, so before we talked about cilia and how they can like produce these wave-like motions, and that can propel mucus up and out of the respiratory system, for example. Also, these little cilia can push along the cerebrospinal fluid, you know, circulating it in the absence of a heart. Also, in specialized parts of the brain, there are ependymal cells that are modified to also produce this cerebrospinal fluid in the first place. Okay, so again, lining this little canal in the middle here are ependymal cells. Okay, so this is a summary of all of those different cell types within nervous tissue. Astrocytes, doing all sorts of different things. Microglial cells, the little Roombas, oligodendrocytes, myelin sheaths, ependymal cells producing and circulating the cerebrospinal fluid within or circulating within the central canal of the spinal cord. In the peripheral nervous system, we have Schwann cells, and also we have satellite cells, which are similar to astrocytes in that they just kind of make a nice cushy environment for the neurons. Okay, so the reason I go over this now is not only because learning about nervous tissue is helpful and will of course be in the test, but also because understanding what makes up nervous tissue, particularly in terms of oligodendrocytes, right, versus cell bodies and synapses, is really important to being able to understand the rest of the spinal cord. Okay, so here is another representation of the spinal cord. As we know, sensory information or afferent information enters the spinal cord here, right, via the dorsal root. Remember that the dorsal root ganglion has all of these neuron cell bodies all grouped together. Synapses with interneurons, okay, and some of those interneurons can immediately trigger a motor response, and that signal travels out and down the ventral root here, okay, via these efferent neurons. But note that all of these synapses, so cell body, axon terminal, right, cell body, axon terminal, all of that is located within the gray matter, within this little butterfly shape right here. And also we can see the central canal. The gray matter is actually organized according to essentially where the sensory information is coming in from and where the motor information is going out to. As we can see here, synapses relaying information from somatic places, so your skin, your muscles, are going to take place here. Here, this is where synapses relaying information coming from viscera, your heart, your liver, your lungs, are going to take place. Okay, so these are sensory nuclei. A nucleus, in addition to being like the housing, the organelle with all the DNA in a cell, in the central nervous system, nuclei are groups of neuron cell bodies, just like ganglia are groups of neuron cell bodies outside the central nervous system. Okay, so again, this gray matter happens to be organized pretty thoroughly. Okay, in this little extension down here, right, and so the bottom of the butterfly wing, motor commands are sent out to the visceral organs from here. Okay, here, this is where somatic information is sent from, right? So again, neuron exiting out the ventral root. Okay, so lots of organization. These are motor nuclei because they are where the dendrites of the motor neurons are located. Okay, we can add names, go figure. In the dorsal or posterior tip of the butterfly wing, where all the sensory nuclei are, we can call that the posterior horn or the dorsal horn. Either one is okay. In the bottom tip or the front tip of the butterfly wing, we can call that the anterior horn. And so that is where, for the most part, the somatic motor nuclei are located. And then this little bump in the middle, which happens to correspond to the visceral motor nuclei, that's called the lateral horn, right? Because it's sticking out actually in the lateral direction. Okay. There are similar names for the organization of the white matter as well. And this is something we're going to get into in a lot more detail at the end of the term. But for now, I just want to point out that right, there are these bundles, these tracts of myelinated axons. So myelinated with oligodendrocytes axons. And so they are essentially going up to the brain or back down. Actually, it's the other way around. These are ascending tracts going to the brain. These are descending tracts coming back down from the brain. Okay. So again, we're going to learn some of these terms later this semester. I just wanted you to be aware that we can name these bundles of neurons within the white matter similarly to how we can name the structures in the gray matter. All of the terms that are blue and underlined here are important for your exam and for your practical. The other ones, the ones in black, are not. Okay, with the exception of the spinal nerve. You should know the spinal nerves as well. Okay. If we take a section, a transverse section through the spinal cord at different places along the spinal cord, the shape of that gray matter versus white matter is going to change a little bit. Right, so there is this regional variation in the structure of the spinal cord, and that's all tied to what, what organs are located in these different areas. Okay, so what I really want to point out is that in the cervical spinal cord, there's pretty much just a ventral root and a dorsal root, or posterior and anterior, horns, right? Posterior, anterior horns. But when we get down into the thoracic and the lumbar, we see a pretty pronounced lateral horn in both of those areas. Okay, and finally, when we get down to the sacral, once again, we see the posterior and anterior horns only, no lateral. And so what's going on there is that in the thoracic and lumbar, that's where we have a lot of visceral organs, right? We need to command the heart. We need to command the lungs to do all sorts of different things. And so in those areas only, the thoracic and the lumbar, do we actually have that more pronounced lateral horn? Whereas in the cervical spine, there's really not a lot of visceral organs, right? Nothing too much, as well as down here by the sacrum as well. Okay. Secondly, the spinal cord doesn't actually extend through your entire spine. Okay. In fact, for the first couple years of your life, in fact, the first 4 years of your life, both your spine and your spinal cord are growing at about the same rate. However, at about 4 years old, your spinal cord actually stops growing. And so in order for the spinal nerves, right, L2, L3, L4, all the way down, to actually exit at the appropriate spot of the spine, the spinal nerves have to grow, right, beyond the end of the spinal cord. And of course, the spinal cord has that butterfly shape within it, the white matter, the gray matter, all the things we just talked about. So that really organized structure stops at about L1, but all of the other spinal nerves have to keep growing to exit here at, right, this would be spinal nerve L3. Okay. because it's under L3. So if you were to take a section through, whatever is in the vertebral canal way down here, what you would see is just a bundle of spinal nerves. And so that bundle is called the cauda equina. Cauda for tail, right? The caudal region is the tail region, equina for horse. So this is called our horse's tail. Okay, the spinal cord, let's take a look at it in the spine. We are looking at the spine from the back, right? So here is the spinous process, transverse process, and up here towards the front is the body. We actually know that this is in the thoracic region because of this costal facet right here. Okay, so that's where a rib would articulate. Okay. Here we can see that this is the dorsal root kind of bulba, or sorry, the dorsal root ganglion, and here the dorsal root. Okay, here is the ventral root, and of course they both join together to form the spinal nerve. Okay, we can see the white matter, okay, with tracts going up and tracts going down. Okay, and the gray matter where all of this sensory information comes in, motor information goes out with the synapses in between. Okay, and this of course is the central canal. Okay, what I want to point out now is that the entire central nervous system, the brain and the spinal cord, are all surrounded by several layers of protective membranes. Okay, so here we can see the most superficial membrane called the dura mater has been sectioned and peeled back so we can see what's underneath. The arachnoid mater actually encloses this cerebrospinal fluid on the outside of the central nervous system, and one final membrane called the pio or pia mater. Okay, collectively, these membranes are called the meninges. Okay, here is a transverse section, and we're looking down into the vertebral canal. Here's the spinal cord, here's the back because that is the spinous process, and the body to the front. We can see 1, 2, and 3 protective membranes, again, collectively called the meninges or meninx for singular. These membranes are there to provide shock absorption. Right, so yes, the bony case around the entire CNS is critically important, but so too is this layer of fat around the spinal cord, right? Your spine is doing all sorts of things, right? It's twisting, it's bending, it's doing all these movements, and housed within it is your ability to control the lower parts of your body. And so we have a lot of fat cushioning and insulating the spinal cord. Okay, deep to this fat is called the dura mater, so dura for durable. This is actually pretty leathery, and we're going to see this later this semester around the brain. It is this dense connective tissue. Okay, deep to that is the arachnoid mater. Right, so that would be right here. Note that the arachnoid mater has this blue space beneath it. Keeping this space open are a series of protein fibers. Okay, and the protein fibers kind of sort of look like spider legs. Therefore, this is called the arachnoid mater, arachnoid meninx, for its appearance to spider legs. So the reason that we need this subarachnoid space, so under the arachnoid mater space, is that this is where the cerebrospinal fluid is circulating on the surface of the central nervous system. Right, so it circulates both inside and outside the CNS. And finally, the pia or pia mater. This is super stretchy, so lots of elastic. This is pretty much right on the surface of the spinal cord. Right, so this here right on the surface, and just, one extra layer of protection. Okay. Again, the entire spinal cord is surrounded by loose adipose and loose areolar connective tissue in this epidural space. So remember, epi means on top of, dura on top of the dura mater, this durable layer. And so when I say epidural, you probably immediately think of the procedure epidural, and I'll show you that in a moment. Okay. As a whole, the meninges maybe are more familiar to you than you think. And the reason for that is that um, in uh, an infection of the meninges is called meningitis, so inflammation of the meninges. There are several different types of meningitis, and I'm not going to go into it in too much detail here, but essentially, some type of microorganism, bacteria, fungus, amoeba, right? So some kind of protist, or even a virus can infect these coverings around the central nervous system. Bacterial meningitis is often the most common, and so that's part of or caused by a strep bacteria. Okay, so here we can see in baby that baby gets strep. All right, so some kind of streptococcus bacteria. These bacteria ultimately can, move into the spine and therefore, try to infect the spinal cord. But what is protecting the spinal cord are the meninges. And so your body responds by producing this inflammatory response, which is, of course, very helpful. But the fact that the meninges are literally like right on the surface of your spinal cord and your brain means that all of that swelling is compressing those nervous system structures. And so this is potentially fatal when the infection gets up to your brain, right? In fact, the gray matter of the brain is on the outside of the brain. And so all of those synapses, all the communication between neurons that controls literally your entire body is right there at the surface that is being compressed by the inflammation in response to the infection. So that's why we get vaccinated against meningitis, because it is fatal. Okay, so the last thing the last thing I want to talk to you guys about is other structures and organization of the peripheral nervous system. Okay, again, I just talked to you about the central nervous system and how the gray matter and white matter are organized in this very predictable way. The entire central nervous system is covered by the meninges and adipose and bone. So lots and lots of protection of those structures. In the peripheral nervous system, our spinal nerves and the subsequent branches of the ventral rami, that of course we're going to name later, they don't have the same levels of protection. Okay, they don't have meninges, they don't have bone, they don't have like fat and everything surrounding them in particular. They are pretty much just like surrounded by muscle, surrounded by fascia. Okay, but they still do have a pretty predictable organization. Okay, so here, if we were to take a section through the spinal nerve here, what we would see is this. The entire spinal nerve is covered by a structure called the epineurium. So again, epi on top of, neurium, N-E-U-R, is a root for like nervous tissue or neuron. And so this is a membrane that is all the way on top of the entire nerve. Right? Within the nerve, there are also blood vessels, right? It's going to take a lot of ATP to generate those concentration gradients and therefore send action potentials ultimately. So we need lots of blood to deliver oxygen and sugar so that our neurons can fire. Okay. Within the nerve are bundles of neurons. Those bundles are called fascicles. And each fascicle is surrounded by a perimeter neurium. Okay. Each individual neuron is, of course, myelinated by Schwann cells, but around that is an endoneurium, so one final layer of connective tissue sheathing. And this is actually something that we've seen on that neuron model in the lab, right? So the endoneurium, that connective tissue sheath that actually surrounds the Schwann cells, which of course surround the axon itself. Okay, so at this point, maybe this seems to be getting familiar. The skeletal muscle is organized in literally the same way. We have a muscle cell, a myofibrous surrounded by an endomysium, multiple muscle cells joined together to form a fascicle surrounded by a perimysium, and multiple fascicles joined together And surrounded to form the entire nerve or muscle, right? The muscle is surrounded by an epimysium. The nerve is surrounded by an epineurium. So the same roots, same kind of nested organization in both the nerves and the muscles. Okay, so that is all for this video. Thank you so much for watching. Stay tuned for the next lessons. Thanks.